Hello, everybody. Thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics related to antibody discovery and development. Our expert speaker, Dr. Sebastian will discuss a new technology for tackling current challenges in early developability assessments with us. Please note the webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and the questions will be answered at the end of the webinar. Without further ado, I'll turn the show over to our speaker, Dr. Jiwing.
Thanks, Kristen, for the introduction. It's a pleasure to be here today. And hello, everybody. Thank you for the time to join this webinar. And I'm going to talk about what we at PAIA do in terms of developability assessments and show you our new technology in this presentation. So a quick look onto the agenda of today. I will start off with a brief company introduction to the company PAIA Biotech, and then I'll dive into the developability section of my presentation, show you what we see as current challenges in the field and what kind of assays are used to measure developability. In the second part, I will explain how our special microplate technology works and why we've find it a very nice and good tool to address these challenges. And in the third part, I will show you quite a lot of data on our developability assays, especially on a map panel of roughly 40 molecules that we have used to characterize those assays. And then I'll, of course, conclude. And if we have time left, I hope I can answer some of your questions. So quick introduction to the company PAIA Biotech. We are based in Germany. We're 10 years old by now. And what we have developed in the past is a microplate technology which allows us to simplify, to streamline so-called bead-based assays. And as you can see from the timeline here on the bottom, we have over the past years developed a number of different assays. We started off with titer assays and then went more into the critical quality space, developing glycan assays, aggregation assays, purity and mispairing assays for bispecifics. And then in late 2023, we went into the developability field and launched a couple of assays there. And this has really taken off in the last 18 months. And this is what I'm going to present today. So basically, we consider ourselves as an assay solution provider for the discovery space. And what does that mean? That means that first we develop assays that we deem important for developability assessments, but we also do custom developments for customers with specific needs. Those can eventually become then also available for the broader community. And I will show you later on two of these assays in more details. So our products in the end are off-the-shelf assay kits comprising a plate and reagents. And these kits you can measure on standard fluorescence plate readers. So not a lot of equipment needed to perform these assays. So let's jump into the developability topic. And as you can see here from this illustration, there are you can see that developability is a really complex topic, and there are a lot of parameters that ultimately influence the developability of a molecule. And what is especially challenging is to predict critical properties, for example, clearance or viscosity, parameters which usually are measured very late in the process, but we will still want to predict it early on with early assays. So this is the first challenge that we see. The second would be that there are no standardized assays and protocols which are established in the industry, which makes it difficult to reproduce even published data. And we also see a lack of miniaturized, automated, and high throughput assays, which are really essential to generate large data sets when you want to do AI and ML. So I will talk today a lot about clearance, and associated with clearance, of course, is polar reactivity. And I think it's important at this point to get the terminology right. So a lot of times people use polar reactivity and polyspecificity, but meaning the same thing. So polyreactivity really is the weak interaction of a molecule with surfaces that are present in the human body. So, for example, cell surfaces, but all kinds of molecules like DNA, all kinds of proteins which you find in the body. So this is really weak interactions that are driven by hydrophobicity, but also by charge. So, for example, if a molecule is positively charged, it may stick to the cell membrane. So this is really polar reactivity. And polyspecificity is something completely different. It is the potentially strong binding of a molecule to a non-related target. So that is rather something impacting the safety of a molecule. So starting into this field, I would like to show you a recent and quite nice example of an attempt to predict pharmacokinetics, PK. And this is from a paper from Adimab and Sanofi from last year. What did they do? They, from an initial yeast screening, they selected 43 maps with quite a broad distribution in polyspecificity or polyreactivity. They chose those maps for production and then also generated PK data in mice for these molecules, and afterwards characterized these molecules further using this 13 different developability assays with the aim to then afterwards look at which kind of assays or combinations of assays are capable of predicting the PK. And as you can see from left to the right, the more assays you feed into the model that they use, the better the prediction of the PK gets. So you see this line here, Spearman correlation of 0.75. This is apparently kind of the goal. And the more assays you use, the closer you get to this quality of prediction. So that's kind of what you could expect, and it's nice to see that this is possible. But on the downside, you see that a lot of combinations of assays include an FcRn assay or other slow and complex assays like CIC, AC-Sins, BVP, etc., etc. So would be nice to get to this kind of PK prediction with more high throughput and easier assays. So what do we have available in the toolbox? Other assays which may be suitable for this purpose. So this is kind of an overview of what we see people using in the field. So there's a lot HPLC based method. Most of them we've seen on the previous slide already. Then we of course have the AC-Sins and the SMP method, which is an effects method that Adimab has pioneered. And then we see a lot of different ELISA assays where people use human antigens to test whether molecules bind to them. We also have the baculovirus particle, which assay, which is pretty popular, and we've seen this in the previous slides. But we also have ovalbumin as a PSR alternative. We do have a DNP HSA, another reagent which was proposed to be an alternative for looking at polyreactivity. So a lot of different assays to consider, and if we want to break down those different technologies and look at the pros and cons that they have, uh, we can summarize chromatography methods as being rather slow, and this is especially true when you look at FcRn, CIC, or heparin chromatography. They need quite a lot of protein, those methods, but on the contrary, they are very reliable. They have defined surfaces on the columns, so it's a pretty robust and good system. If you're looking at ELISAs, then you see that a lot of these ELISAs which are used in the fields use negatively charged antigens, which leads to quite a high correlation. You could also say redundancy of assays, so very high correlation of those assays. They need quite a lot of material as well. And of course, in ELISA, everybody knows there's a quite pretty complex workflow. And one thing which I'll discuss later is that BSA is also used as a surface blocker in these assays, which is kind of the common characteristic that it shares with FACS methods, where you also have BSA. You're using pretty complex reagents, which can make problems because you may have batch-to-batch variations. And of course, the FACS machine is also a rather complex machine to run. So I would like to in the next slides, talk about the topic of using BSA. There are also people around who say that they don't need to use these ELISA assays because they correlate a lot to BSA anyways. So the question is, what do we really measure in these assays when we use, for example, insulin ELISA and block with BSA? So this was a question which was not really discussed until last year when this paper from Adimab appeared. No, not Abimab, it's AbbVie, sorry. What did they do? They looked at what is happening in a BVP, baculovirus assay, and they compared the results for BVP assay with a BSA assay, and they got for like 200 molecules, I think, this kind of scatter plot here, and wondered what is happening here. And then they discussed this assay in more detail, and especially the BSA blocking that they use in the BVP assay, and came to the conclusion that what you get in those assays really is a mix of interaction. We know that the baculovirus particles have a strongly hydrophobic surface. So what we measure in these assays in the end is not a single thing, not only the BVP, but it's a hydrophobic interaction from the BVP in combination with a charge-based interaction, which comes from the blocking with BSA. So that's interesting. And the second interesting thing that they found is that concentration plays a big role in these ELISAs. For example, they found that only roughly 50% of the molecules in the BVP ELISA were polyreactive at both of the concentrations that they tested. And in the BSA setting, it was even worse. Only 18% showed the same kind of behavior in this assay. So as a conclusion, they say that really you need to pay attention to the concentration at which you're running those assays, and especially if you want to feed this data into models. So the question is really, what are we really measuring in these ELISAs and in the BVP setting? This it's important to keep in mind when we're talking about the BVP assay that we are proposing at PAIA. So I also wanted to touch on the topic of reproducibility of assays, and this is two two graphs taken from the Jane paper from 2023, where they are comparing the results of two labs running the same maps on a Higg column. That's what you see here on the left-hand side. That's what we all want to see, almost perfect correlation of the samples. And on contrast, on the right-hand side, you see PSR method compared to an assay, also a soluble membrane protein-based assay. Here, two different labs again, and of course you see a lot more variation in this plot. And another conclusion from this paper is that you cannot expect a quantitative agreement in these kind of assay settings just because the reagents are very complex, and also the workflow of the assay is pretty complex. So that in the end, you need to run additional assays to make sure you're not missing something in your developability assessment. So to sum up this first part of the presentation, complex reagents and assays may lead to unreliable data, and then you need to run additional assays. In contrast to this, the data from chromatography is really very reproducible and robust, so everybody trusts these data. But oftentimes we're using a lot of, or at least several quite similar, often ELISA assays for flagging molecules. And usually what people do is you flag 10%, 10% of the highest binding molecules in an approach to really de-risk the selection of lead candidates. But this, in the end, does not really answer the question if the molecules are good and bad, because this is kind of an arbitrary thresholding that you introduce, and this is, of course, depending on the cohort of samples that you are running. So what we ideally want is to have an assay set that lets you predict those critical quality or critical parameters for PK and viscosity. We want to have fast and reproducible assays. We don't want to spend a lot of material on those assays because we want to, look at different parameters or a lot of different parameters ideally. And it would be also nice if those assays that we have are so precise and defined that they allow to get some mechanistic insights for what is really going on in this assay, so that we can feed this into models. And we at PAIA think that with our technology, we can contribute to this goal. And this is leading us to the second part, which shows you how our microplate technology works. And as I said in the very beginning, we use this special plate technology to streamline bead-based assays. So what we want to do ultimately is we want to measure the binding of an antibody to a bead with a certain functionality on the surface. And in order to do that, we use an Fc-specific nanobody with a fluorescence label attached to it, and we then really want to measure whether that binds to the beads or not. So we're performing these assays in special kind of microplates, and you see a well here. We're starting off with the beads. They are coming in the plate already, and you can see that this plate has a pyramid here, and this pyramid is transparent, while the rest of the well is completely black. So we start adding the reagent containing this nanobody to the well and the sample. We place the plate onto an orbital shaker for half an hour. And as you can see, the reaction takes place here. So the antibody and the nanobody bind to the beads. We then stop the shaking so that the beads can sediment, and you can see here that the pyramid is directing the beads to the sides and the bottom of the well. And then in the last step of the workflow, we measure the fluorescence from the bottom on a plate reader. And you can see that this pyramid does two things. As I already said, the beads are directed to the sides on the bottom. And also this pyramid serves as a measurement window so that when we measure the fluorescence from the bottom, we only measure the fluorescence which is left here in the solution. So whenever we see binding, we see a drop of fluorescence, and that's important to keep in mind. So low fluorescence in our assay always means that binding has taken place. So this is a pretty straightforward assay workflow, which allows you to run a whole 384 well plate in less than an hour. And if you combine the whole setup to the other methods that are out there, you will see that it's pretty similar in the end to chromatography in the sense that we have the same kind of surface that we're using. The only thing that we add is this nanobody, and of course we don't have a flow system but a static system, but otherwise pretty simple in terms of components that we use. If you compare that to ELISAs or fax technologies, yeah, it's really seems minimalistic. And the other thing is we also don't use huge amounts of BSA, which is what is used in ELISA and fax-based methods. So what do we have available on our platform as of today? This is an overview of the assays that we have for the Fc-containing proteins. So we have 3 assays here: heparin, cation exchange, and hydrophobicity, to replace the slow HPLC methods. And on the ELISA side of things, we have an assay to replace BVP assays, which we call this CDBVP mimetic. CD stands for chemically defined. We have an albumin assay to look at polyreactivity as an alternative to PSR, and then we have human albumin and dinitrophenyl human serum albumin as well, which is, well, maybe a little bit of exotic assay, but it's also been proposed to look at polyreactivity. So that's the assay portfolio for the Fc-containing proteins. And moving on to the HISTEC proteins, we have less assays available, but we have a heparin assay, we have a hydrophobicity assay, and here for polyreactivity, we have the albumin assay. So we hope to add some more assays to this portfolio for HISTEC assays, in the next 1 to 2 years. Okay, and with this, I would like to transition to the assay data that I want to share with you today. And I would like to start off with the, what we call the chromatography-like assays, because they use exactly the same kind of material that you have on your HPLC column. So it's exactly the same kind of resin. So if you look at those plots here on this slide, it's important to keep in mind that in the chromatography, binding relates or correlates with high retention time. So these molecules that you see here are the ones that binding to the column. And in our assay, binding shows as low fluorescence counts. That's what you see here. So you will always expect a negative correlation between retention time and our fluorescence readouts, and that's what you see in all 3 plots here. So exactly what you want to see. And what is important to mention in these assays is that these assays are pretty independent of map concentration. So you can run these assays in concentrations of 5 to 200 micrograms per mil of antibody, and you will still get the same kind of results. Very robust in terms of map concentration. And what we show here is the results for 10 microgram per mil. So pretty little material consumption. So chromatography is always easy to evaluate also for customers because yeah, the correlation is always great. And things become a little more tricky when you're looking at polyreactivity, because there, yeah, the assays that you want to compare with are less robust than chromatography. So looking first at the polyreactivity assay using the ovalbumin, a little bit of background. Ovalbumin is a molecule that was proposed by the lab of Peter Tessier as an alternative to, yeah, soluble membrane protein-based assays, because the molecule is having a low PI. It carries several post-translational modifications. So there's some potential for different kinds of interactions with an antibody. And what we did to validate this assay and to confirm that ovalbumin is a good alternative, we developed an in-house developed assay with soluble membrane proteins attached to the beads. And what you can see here in this plot is a very nice correlation between this SMP-based assay and the ovalbumin assay. So we see the same kind of binders here on the lower bottom here. So nice correlation, and also here on the other end, the non-binders agree in both assays. So pretty convincing results. And the other polyreactivity assay that we have in our portfolio is this already mentioned chemically defined BVP mimetic assay. And the starting point of this development, this assay development, were really discussions with our customers who asked us whether we have something in our portfolio to replace the traditional BVP assays, because it's really challenging to get good results with this assay. So it also posed a problem for us in the sense that we didn't know which kind of assay data is really reliable in terms of BVP assays. So what we did is we asked our customers for their BVP assay data in order to identify a reliable set of maps for which we know that they're really binding or not binding. So in the end, we came up with this list of antibodies here. And you can see that we have here 4 antibodies which are flagged by our customers as well as in the Jane paper. Then we have the denosumab, which is a kind of special case. And then you have some molecules in which it's not clear from the data whether they are supposed to be BVP binders, yes or no. And then you have a whole list of molecules down here which are non-binders. So what we then did with this set of molecules, we screened different so-called multimode chromatography resins to see which ones gives us a similar reading than those BVP assays. So multimode resins are resins which have 2 different functionalities. On this bead surface, there is some hydrophobicity on the beads, as well as some cation exchange function. So it's really a combination of effects that you see presumably in these traditional BVP assays when you run them BVP plus BSA coating or blocking. So, and this is the data that we got with this one multimode resin that we finally selected. And you can see that this assay binds those 4 which have a high BVP ELISA score. So scores also um, um, scores as a binder in our assay, so low fluorescence intensity. And then we have the denosumab, which is a special case because in the final drug format it's an IgG2, and there it's a non-binder. If you reformat this molecule to an IgG1, then it apparently becomes a BVP binder. So that's also quite a nice illustration on how tricky these things can be. And then, of course, we have the whole crowd of non-binders here, on which the 2 assays agree. So this is what we have in terms of assays for FC-containing molecules. So now let's transition over to the assays that we have for the his-tag proteins. And this here is the first example. That's the hydrophobicity assay that we developed in cooperation with Sanofi. And this project had two stages. In the first stage, we got 9 reference molecules from Sanofi together with the retention times that they had measured for those molecules. And we developed an assay which gave us this pretty linear relationship. Again, here, correlation of high retention times with low fluorescence counts. So the same thing that you've seen on previous slides. And with this kind of assay, we then went into the second phase of the project, in which we were given 3 different blinded sets of molecules at different stages of purification. So we had 1-step, 2-step purified proteins in there, and then crude supernatant. And as you can see here, we again get a very nice linear relationship between Higg retention and our assay response in a range of about like 7.5 minutes to maybe 12, and then it's leveling off here. So the very high the molecules with very high hydrophobicity really come as a pack here at the end of this plot. So the only sample preparation that we had to do for these sample set was simple dilution in water, so you can even use these assays in crude supernatant, which is also maybe quite nice for some customers. That's the Higg assay for the his-tag proteins. And the second one is the ovalbumin assay. So we're looking here at polyreactivity. Another project which we did with pharma, and kind of the setup of the project was pretty similar to the previous project. Here we got different his-tag fabs from well-known full-length antibodies. You see the list here, and those were selected in such a way that we have some molecules in there with high polyspecificity, with medium and with low polyspecificity scores from the literature. So the aim was to look whether these his-tag fabs show the same kind of behavior as the full-length antibodies. So we went on to develop the assay. We again use ovalbumin-coated beads, but of course we changed the reagent. In this case, we're using a 6 his-tag specific reagent, also of course with a fluorophore attached to it. And then we looked at whether or how well this 6 his ovalbumin assay on the fragments correlates with the FC ovalbumin assay performed on the full-length IgGs, the corresponding ones. And you see a very, very nice correlation of these two assays, which kind of underlines what we think to have understood that most of the polyreactivity comes from the fab, and there's not a lot of influence of the FC part, because you see this correlation between these two assays so well. And then, of course, the other thing that you look at is looking at whether the results from this 6 his ovalbumin assay correlate with the published data for the full-length antibodies. And again, you see nice correlation with what you would expect. So the binders in this assay correlate with the molecules that show high PSR scores in the Jane paper. So exactly what you would expect, pretty straightforward results here. So, and coming closer to the end of the presentation, I would like to give you some impression of how what kind of data you can obtain if you run several of our assays, and how to interpret these assay data. So here you see a typical set of 30 different molecules that were run in 4 different assays. And you see that for a lot of samples, you see quite a strong correlation of the CEX, ovalbumin, and heparin assay, because they are negatively charged beads used in this assay. So here they are all red here in the highlighted cells here. And then the HIC quite often is showing the opposite trend, but there are also exceptions. So yeah, usually quite some correlation between those negatively charged assays, but you can also see exceptions or differences in the different molecules. For example, if you look at number 21 here, you can see there's no CEX binding, so no binding to negatively charged beads, but quite strong binding to heparin. So you can say for this molecule, this is not ionic interaction. This is an interaction with heparin, which is not um, um, uh, based on ionic interaction, right? And you can have this in all different flavors, and we're really curious to see how what kind of effect these data sets have once you feed these data and these mechanistic insights into AI models and what you can get out of those. And then last thing, of course, that you can do with this assay, and that's maybe the most easy thing, you can also spot the good molecules very easily, because these are those who don't show binding in any of those assays. So whenever everything is more or less blue, that's a good sign, and those molecules will be good in terms of developability. All right, so some practical considerations, or just to show you our technology at one glance, what comes with it, and how much work you have to put in to get this running. So we usually say that you need to invest like 3 to 4 days max to set up the method in your lab. That means some housekeeping tests on the equipment that you have. And once this is running, you really only need approximately 1 microgram of protein per sample to run an assay in triplicates. You can achieve a throughput of hundreds of samples a day, even without automation. The shelf life of the assay kits, which is a plate essentially, and the reagent is 6 months. And what is important to mention is that you can also only use a part of the plate if you don't have a sufficient amount of samples. And if you run those molecules, for example, in triplicate, you can expect pretty decent CV in the single digit range, typically around 5 to 7%. And of course, you get a whole lot of data out of these kind of measurements. And the price tag is like $1.50 to $2 per data point, to give you an idea about this. Yeah, and with this, I would like to wrap up this presentation. I hope I could show you that we really have a nice suite of assays available with very simple workflows, very defined beads and reagents, and these are really nicely suited for doing developability assays. They allow you to run a lot of samples really quickly, and to generate really large data sets. In terms of automation, is pretty straightforward as well. And the good thing is that once you have established a protocol for one assay, you have one protocol for all the assays. And yeah, we've seen really excellent correlation with published data. And with this, I would like to conclude the presentation. Thank you for your attention. And I guess we have some time for questions and answers. Thanks.
Thank you so much, Dr. G. Ring. So the first question is the shelf life of an unopened, unsealed plates 6 months or after opening?
Yeah, excellent question. So the shelf life of 6 months is for the unopened, but after opening, it's good for another 3 months.
Thank you so much for your question. And audience, just a reminder, you can add additional questions to the Q&A section if you have any. Um, Another question for you is, have you considered developing assays for smaller or larger antibody concentrations outside of the 5 to 200 microgram per milliliter range? Or is that something that's feasible? Or would you run into some type of complications by trying to go outside of those concentrations?
Yeah, also a great question. So we know we can go a little lower than the 5 that I mentioned in in the presentation. I think we're a little bit limited by the amount of fluorophore that we need to be able to detect, but we know we can go down to like 2 or 3 microgram per mil, but much lower, yeah, would be maybe a bit complicated, but or would at least require us to develop a different kind of detection.
Gotcha. Okay. Thank you so much. And then, what are some future directions that you have for a continued study of these developability assays?
Yeah, I mean, we are always looking to add new assays in the future. Of course, I mean, we of course would like to have something which is going into the direction of self-association. This is something that we can't do currently yet, but we're thinking about it, of course. And then, of course, FCRN would be a huge thing. So stay tuned. We're working on these topics.
Wonderful. Okay, we have a couple more questions in the chat. Why, in the case of the his tag protein, you use the term polyreactivity and not polyspecificity?
Yeah, that is one question that I addressed very early in the presentation. So polyreactivity is the term that we are using, and this was also something that was proposed. But I know that people use those terms interchangeably. So polyreactivity for us means really weak interaction with all kinds of molecules and surfaces that are in the body. And polyspecificity is really a completely different thing, is when a molecule is binding strongly, potentially, to a completely unrelated target. And for those kind of analysis, you need like protein arrays where you exactly know which kind of, for example, receptors you bind to. That is polyspecificity.
Perfect. Thank you so much. And then the final question, unless anybody else wants to drop anything else in the chat, is there a particular plate reader that you recommend?
Well, yes. Well, we have our favorites, but usually, and with a little bit of tweaking, all the main brands and manufacturers work. So I could drop in some names here, but really all the big brands have are compatible with these assay plates. They are standard plates. It's a standard grinder frame that we use, and as long as these readers can do bottom reading fluorescence, that's all good.
Perfect. Okay, now we do have a couple more. How reproducible are the results for this assay? Can the specific values be reproduced to facilitate AI models, or is it simply whether or not it passes the polyreactivity threshold?
Yeah, so excellent question. The thing with the thresholds, we don't really like thresholding, right? Because this is kind of an arbitrary line that you're drawing somewhere. We want to have reproducible assays where you can really compare one-to-one the raw intensities that you see on a Monday, on a Friday also. So they are very reproducible, those assays, and we want to not take flags into some models, but real numbers, so quantitative data. I hope that's answering the question.
Perfect. And the last one, I think this is just to reiterate the shelf life of the reagent.
Yeah. So it's 6 months is the shelf life, and after opening the kit, it's another 3 months.
6 months and then 3 months. And is it possible to multiplex?
That's also a pretty common question. Unfortunately, no. So we're running all the assays with the same wavelength, but it's only one wavelength that we're using, and that's in the red region. So excitation is 630, 670 emission, to give you an idea.
Perfect. Okay. This similar clarification about the shelf life. So the shelf life of both the plate and the reagent that comes in the bottle are both 6 months.
Yes. Yes.
Okay. That was the last question in my queue. I'll give everybody just a couple more. Oh, every time I say that, another one comes in. Are the plate dimensions available to allow for liquid handling automation?
Yes. Yeah. As I said, this is a standard grinder footprint, like a 384 flat bottom plate. So it's really absolutely standard, and a lot of people have automated this already.
Perfect. Okay. I'm not seeing anything else come through. So if that is the end of the questions, I will go ahead and wrap this up. Thank you again for this insightful presentation, Dr. G-Ring. The on-demand version of this webinar will be available very soon, and a link will be sent by email to everyone who registered. Feel free to watch this or any of our other on-demand webinars when it's convenient for you, and this will also be available on the Antibody Society's YouTube channel. Thank you again, and have a great rest of your day.